So I'm John Gribben. Welcome to IWNHL here in now sunny Cambridge, Massachusetts. We've had a great day again today, discussing lots of topics related to non-Hodgkin's lymphoma. Fabulous overview from what happened in the real classification from Elaine Jaffe, a real retrospective back look at how we got to where we are with our current classifications of lymphoma, which I'll come back to talk about in just a moment. Wonderful session looking at some novel therapies and where they fit, and then ending up today with a really good session on CAR T cells and other immunotherapy approaches, which of course are hugely exciting in this field. But right before the CAR T cell session, we had our keynote address from Margaret Shipp, who needs no introduction to in the field of non-Hodgkin's lymphoma, and Margaret's made her whole career out of identifying initially the clinical and now the genetic patterns underlying the prognostic features of non-Hodgkin's lymphoma. So I'd say you're the queen of prognosis.
I don't know about that term.
So you gave a phenomenal talk. I mean, the issue, of course, of what you were talking about is the heterogeneity that we see within diffuse large B-cell lymphoma. And you said it yourself that we thought we'd made major advances in terms of identifying the putative cell of origin, and that by identifying targets that were putatively important in that, we would be able to see advances, particularly for the poorer prognosis ABC lymphoma. And of course, that wasn't what we saw, right?
That's true. That's true.
So you've taken the approach now of going back and looking at that data in a slightly different way, looking at using more modern technology to integrate more information than we have from just the gene expression profiling studies, and identified various clusters that behave in biologically similar manners that don't quite define exactly the way we thought of in terms of ABC versus GCB, right?
Right. So if you think about it, and even going back a little bit before we had the information about transcriptional profiling, diffuse large B-cell lymphoma, which is the most common lymphoid malignancy in adults, we've known for a very long time that it's a very heterogeneous disease, both from a clinical perspective and a molecular perspective. And so there's been a great deal of interest in trying to understand that heterogeneity, with the idea that understanding that heterogeneity would help as a way forward in terms of developing better treatments. And
I thought actually Elaine set that up very nicely with her talk earlier. Mm-hmm. The whole approach that the histopathologists were taking, even though the clinicians didn't appreciate it at the time, has actually helped us enormously to start to think about these diseases in exactly that way. How do we divide up the pie to make it be a manageable size that becomes a homogeneous group to think about?
Well, or to say that in a slightly different way, it's recognizing heterogeneity.
Yes.
So you can, and I think what Elaine was talking about was recognizing heterogeneity in the sense of discrete lymphoid malignancies, diseases that we had not appreciated as being different diseases in the past. But with regard to diffuse large B-cell lymphoma, I think we've all thought for many years that there was heterogeneity that you couldn't appreciate by looking at the cells under the microscope. So that was really the idea. We've had clinical prognostic factor classifications that were robust, really helped us identify patients who were more or less likely to do well with standard therapy. But one of the challenges is that even though they were helpful in terms of identifying patients who were more or less likely to do well, they didn't tell you anything about biology, nor did they give you insights as to how you might treat more effectively. So that's where the hope is that biology and genetics would be more helpful. And cell of origin clearly represented an important advance in terms of identifying heterogeneity not appreciated by looking at the cells under the microscope. Two broad categories of lymphomas, GCB, ABC, a suggestion that ABC did less well, so that might be a group that you would target for experimental therapies. But as you mentioned, the clinical trials that have been implemented to try to improve on the outcome based on ABC as defined transcriptionally have all been negative. And so it really suggests we need to understand whether there's more heterogeneity than previously appreciated. And so that's what our goal was to really understand that. And the take-home message, I think, is that in what is the aggregate of diffuse large B-cell lymphoma, there are, broadly speaking, 5 discrete genetic clusters that have a combination of genetic alterations that really are associated with unique biology and also with outcome differences. And within those 5, there are 2 that have the transcriptional features of the ABC type DLBCLs that actually are quite different in terms of their genetics and also their outcome to clinical therapies. And similarly for GCB type DLBCLs, 2 different variations of that.
So
and
too simplistic to say that ABC is bad and GCB is
no,
good.
No, there are 2 different types of ABC. One that is far more favorable than the other. 2 different types of GCB, one more favorable than the other. And another type that is really independent of cell of origin, so that if you didn't actually understand the genetics, this would not be a type that you would in any way capture with that profiling. And so the hope is that having identified this heterogeneity and having associated this heterogeneity with what we think are meaningful differences in outcome, it will provide the beginnings of a roadmap for thinking more specifically about discrete groups of diffuse large B-cell lymphoma that would not otherwise be identified unless you unless you analyzed the genetic signature and understood these discrete genetic clusters.
It does mean, of course, that we're going to have to do collaborative studies going forward to get the sorts of numbers you'll need to get robust answers when you're taking a disease. Even the most common lymphoma split into these different groups lands up being quite a challenge to be able to identify and move it forward.
So to follow up on that, I think that, and you asked a very good question in the context of the session in terms of how do you capture that information in a way that is clinically useful, essentially. So what's the method that you use to capture the genetic signature, and then how do you classify tumors using that? And so we're working very hard to develop approaches to be able to do that with a much more simplified targeted assay, but a targeted assay that actually captures all of these important genetic alterations.
Do you think you'll be able to do it on paraffin blocks?
Absolutely. We did it on paraffin blocks for the original whole exome sequencing.
Hugely important nowadays where the lymphoma, unless you're going to have to re-biopsy a patient again when they come, but the biopsy's already taken in paraffin.
It can be done in paraffin blocks. And then the other thing that's important for many of the earlier sequencing studies, one of the requirements was that you had to have a normal source of DNA from the same patient to be able to do the analysis. And so we now have an approach that allows us to analyze the tumor samples without having a paired normal DNA. So that also huge advance for taking something into the clinic where it's in many cases less likely that you would have that additional paired normal sample, and also being able to, as you mentioned, analyze the paraffin blocks rather than a specially prepared type of biopsy specimen is also important in terms of making this practically useful for clinical application.
So we've got on the horizon the potential to get a paraffin block from a patient, do an analysis that will tell us much more accurately what are the potentially best targets that patient should have to optimize their outcome. Then, of course, do the clinical trials in that setting. And then we'll have to wait for the follow-up to occur. The issue, of course, becomes, how do we select, and are you going to need multiple targets into those types of therapies? Because even though your work has identified pathways which are often perturbed, you may also need to hit more than one pathway in each of those clusters to be able to see the kind of step advance that we're looking for.
And I think you're absolutely right, because I think one of the things that the genetic analysis has shown is that it's not simply a single genetic alteration or even two genetic alterations in a different cluster, but it's a combination of genetic alterations. And when you look carefully at the combination of genetic alterations, what you can actually see, looking at the data, is that it's genetic ways to perturb multiple complementary pathways, all to the advantage of the tumor cell survival.
Sure.
And so it's an important lesson in terms of our thinking about how targeted therapy might work in specific tumor types. And in the same way that we found that combination chemotherapy was essential, that one single agent was likely to be ineffective and that you really needed combinations of chemotherapeutic agents to work, it may very well be that as we think about the biologic pathways that are being perturbed by these genetic alterations, that when we think about targeting them, we'll be targeting combinations of pathways that are clearly synergistic in a beneficial way to the tumor.
Of course, the other thing is we started this whole conversation saying the clinical trials had failed, but the other thing that your data has provided to us is an explanation that it may have failed for not the reasons that we thought, that is, that the agent wasn't effective, but we were using the agent in the wrong patients. So there is the potential for you to go back and look at those clinical trial samples again and see whether you can find out that particular clusters did perhaps respond to the addition of ibrutinib or lenalidomide or one of the other therapies that have been explored. Do you think that's an advance, or do you think we should just move forward and do the next studies?
So I think that that's a very interesting point, and I certainly think it's worth considering, because I wouldn't say, I think failure is a very strong word, so I would say that the trials did not meet their therapeutic endpoints, and so then the question becomes, why did it not? Why did trials not meet their therapeutic endpoints? And is there additional information and understanding about heterogeneity that would allow us to revisit the trial population in clinical trials like that, to understand whether the trial might not have met a therapeutic endpoint for the entire population, but now I think we have information to suggest that that trial population was not as homogeneous as was perhaps initially anticipated. And so if you can, within that population, identify heterogeneity and identify the subset of patients for whom potentially the targeted agent may be more beneficial, that would be quite important.
So there we have it in a nutshell, that we've identified, or your work has helped identify, and that of others, a lot of the molecular basis of the heterogeneity that we see in diffuse large B-cell lymphoma. That's half a step towards being able to design the definitive clinical trials to address whether, by hitting, as we all expect, the fundamental pathways that are disturbed in the cancer cell, we will and should be able to improve the outcome for our patients.
I want to make just one other point, and that is that increasingly, as we are doing these kind of studies, it's really science as a team sport. And it's really something that requires multidisciplinary groups of investigators who bring to the investigation different skill sets in terms of computational biology expertise, genetics expertise, disease-specific expertise, translation into the clinic expertise. And I think that as we move forward, increasingly, appreciating the multidisciplinary nature of investigation and how that will be important to actually make these clinical advances is something that I think about a lot.
Sure, absolutely. But it does feel as if all the pieces are falling into place for us to be able to at least address the hypothesis with a high probability of success.
I think so.
So there we have it. No huge advances in being able to tell you that we've cured your lymphoma now, but huge advances in being able to think about how we can design future clinical trials to improve the outcome for our patients. I think another really successful day for IWNHL, and we're looking forward to another full day tomorrow to see what other advances we can see in the field of non-Hodgkin's lymphoma.